Guys, and welcome back to my channel. In this video, I'm going to be going over gas transport, which is one of the specific final topics in the respiratory system. In this lecture, we're going to go over basically how oxygen and carbon dioxide are transported in our blood on hemoglobin and just in the blood in general. And then I'm going to record a second video that explains acid-base balance and what it has to do with the respiratory system. So these two videos will be kind of the last bit of the respiratory system that I want you to see. Okay, so we're going to get started with these slides right here. So this is going to be about not even 10 slides or so, just on gas transport. And you guys already know that oxygen enters our bloodstream through our lungs. We talked about this in class. And carbon dioxide leaves our bloodstream through our lungs. But we want to look at what happens with these gases when they're actually in our blood. So how are they transported from our lungs? How is oxygen transported from our lungs to the systemic tissues in our body? Like, is it all bound to hemoglobin? Is some of it just dissolved in the blood? We kind of want to look at how exactly it's transported and how hemoglobin helps out with that. So you can see the definition of gas transport right up here, and let me get my pen going. Okay, great. So we can see that gas transport, this is process of carrying the gases from the alveoli to the systemic tissues and vice versa and back, okay, from the tissues to the alveoli. So if we look on this slide, we can see that oxygen is mostly transported bound to hemoglobin. Okay, most of the oxygen in our blood is actually attached to hemoglobin. We're going to talk about how that works coming up here. And only a small percentage of the oxygen is dissolved in our blood plasma. But it is important to know that we do have a little bit of oxygen dissolved in the plasma, just like we do carbon dioxide that we'll see in a moment. So I want to remind you about the structure of hemoglobin. And I know we've talked about this before, but if you remember, hemoglobin has 4 subunits. So I'm just going to draw 4 circles like this to represent each of those 4 subunits. And you might remember from the blood chapter that they're called globin proteins or globin chains. So each of these subunits is one either alpha globin or beta globin molecule. And think back, what's in the center of each of these molecules to hold the oxygen? Just think back from lecture. What is actually in the middle here that holds the oxygen molecules? You might remember that it is a chemical group called heme. Heme. Okay. And there's one heme group in the center of each of these. So I'm just going to draw a blue square in the middle here to represent that. One heme in the middle of each of these subunits. And part of the heme is a ferrous ion, and actually it's an iron. If you remember, a ferrous ion is an iron ion. Fe2+ is the symbol we use. And so there's one of these ferrous ions attached to each of these heme groups in the center of each of these little subunits of this hemoglobin molecule. So this whole thing that we've got here, this is hemoglobin. And you might remember we abbreviate this HB. Okay, so we've got 4 globin subunits, each with a molecule of heme that's carrying a ferrous ion. And those 4 subunits together with those heme molecules make up hemoglobin, this one molecule of hemoglobin. The way the oxygen is actually carried on the hemoglobin is through this, let's use red actually, is through this iron ion here. So the oxygen is actually bound to the iron. I'm just going to draw it kind of sticking out like this simply because my diagram isn't big enough right now, but it would be in the center of this molecule. Okay? Each of those oxygens is bound to one of those iron ions, one of those ferrous ions. So really, our hemoglobin molecule is going to look something like this, where we have our 4 subunits. And remember, these are polypeptide chains that are very nicely coiled, so we would see lots of coiling going on here. And in the middle of each one of these, we would have an iron and an oxygen molecule. So we can just draw this like this to represent that. All right, cool. So we're going to look at this a little bit further on the upcoming slides. But for right now, let's discuss carbon dioxide. Okay, so for carbon dioxide transport, 70% of our carbon dioxide is actually transported as something called a bicarbonate ion. I don't know if you've heard of this or remember this. You probably heard of it before. But the bicarbonate ion is HCO3 minus. This is the symbol or the molecular formula we use. It's a hydrogen, a carbon, 3 oxygens, and it has a negative charge associated with it. So this is HCO3 minus. And we're going to look at exactly how this works coming up here. Okay, but most of our carbon dioxide in our blood is actually found in this form. So I want you to memorize this ion. A small percentage of about a quarter of our carbon dioxide, 23%, that's not so small, I guess, right, is actually bound to hemoglobin. So some of the carbon dioxide has the ability to bind hemoglobin. And the way it binds to hemoglobin is not the same way that oxygen binds. It's not right in the center of the molecule attached to the iron ion attached to the heme. That's not where the carbon dioxide binds. Okay, the carbon dioxide is going to bind at some available positions on the polypeptide chains here. Okay, so some of the amino acids in the globin chains, in those polypeptide chains, actually can support the binding of carbon dioxide. Some of the amino acids actually allow carbon dioxide to bind. And those sites are different than the binding site for oxygen. So carbon dioxide and oxygen are not directly competing for binding to this little heme group in the middle here. Okay, so these two gas molecules, they're not directly competing with each other. They do affect each other's ability to bind, but they don't bind at the same place in the hemoglobin molecule. All right, we're going to talk about the significance of that coming up. So I really want you to know they both bind to hemoglobin, but they can just bind in some different positions. And then finally, for our carbon dioxide, 7% of it is just found dissolved in our blood plasma, just like we have this 1.5% of oxygen dissolved in our blood plasma. Okay, so on this slide, we can see what we have here is called the oxyhemoglobin dissociation curve. And I want you to be very familiar with this graph. So I want you to know the name of the graph, what the graph looks like, and I'm gonna kind of talk you through it here and tell you about what kind of information it's giving us. So the oxyhemoglobin dissociation curve describes the binding of oxygen with hemoglobin. Okay, it describes how much oxygen is binding to hemoglobin. So I want to illustrate this for you. Okay, so down here, let's draw a hemoglobin molecule with our 4 subunits, just like we just had on our last slide. Okay, and we said there's an oxygen molecule that has the potential to bind in each one of these 4 subunits. Okay, so these individual subunits, they operate independently. Each one can bind oxygen on its own, and they also cooperate with each other. Hemoglobin exhibits a property we call cooperativity, where when one of these 4 subunits binds oxygen, okay, let's just say this subunit right here binds oxygen. When that one subunit binds oxygen, it actually makes the next subunit more likely to bind oxygen. It causes this subunit here to have an even higher affinity for oxygen. And once these 2 top subunits bind oxygen, an oxygen molecule each, they cause the 3rd subunit to have an even higher affinity to bind oxygen. So basically, we're increasing the affinity for oxygen each time a new subunit is binding an oxygen molecule. So once 3 of these have an oxygen molecule bound, that will greatly increase the affinity of the 4th subunit to bind oxygen. So what happens here is that instead of each of these subunits on their own, each having the same likelihood to bind oxygen, just one after another, once one binds, the other has an even higher likelihood of binding oxygen. Once 2 are bound with oxygen, the 3rd has an even higher affinity for oxygen. And once 3 are bound, the 4th has an even higher affinity for oxygen. Affinity is just the tendency for a molecule to bind another molecule. So if hemoglobin has a really high affinity for oxygen, it really wants to grab an oxygen molecule fast. If a molecule has a low affinity for another molecule, a low binding affinity, it's not really that interested in binding to that other molecule. Okay. So this property of cooperativity, where one subunit binding oxygen increases the likelihood and increases the affinity of the other subunits to bind oxygen, this is illustrated by this graph over here. This is our dissociation curve. So let's just look at this so you can get an idea of what's happening here. So on the x-axis, we have partial pressure of oxygen. Remember, that's the pressure due to oxygen being present in the blood. It's the pressure it exerts on the vessel walls. And we're measuring this in millimeters of mercury. Okay, so we have from 0 millimeters of mercury, this would be no oxygen in the blood, up to 100 millimeters of mercury of partial pressure of oxygen in the alveoli. This is pretty much the highest the partial pressure of oxygen will ever be in the blood. And then over here on the y-axis, we have percentage oxygen saturation of hemoglobin. Okay, so what that means is that, let's say we have if we look over here at our hemoglobin molecule, let's say we have a 25% saturation of hemoglobin with oxygen. This means 25%, right? It's like 1 out of 4. This means only 1 of the 4 subunits in the hemoglobin molecule is binding oxygen. So maybe we only have oxygen here. Okay. And if we have 50% saturation of hemoglobin with oxygen, that means half of the subunits are binding oxygen. If we have 75% saturation, this means 3 out of the 4 subunits are binding oxygen. And if we have 100% saturation of hemoglobin with oxygen, this means that all 4 subunits are bound with oxygen. So this is basically telling us the percent saturation of hemoglobin with oxygen. How saturated is hemoglobin with oxygen? Okay. Is it fully saturated? Is it half saturated? Is it a quarter saturated? That's what that's telling us. So over here on the x or on the y-axis, you'll notice that we have other values, right? We don't just have 25, 50, 75%. We have 20%, 40%. Here's 50%. Here's 60%. So what this means, let's say we have 60% saturation of hemoglobin with oxygen. This means if we were to average the saturation of all the hemoglobin molecules in the blood in a particular area, some of those molecules would be at 50% saturation, some of those hemoglobin molecules would be at 75% saturation, and the average would be 60% saturation. Okay, so when we say 60% saturation, it's just because we're talking about many molecules and we're just averaging their saturations. Okay, hope that makes sense. So please let me know if you have any questions. Feel free to email me, or you can comment on the video and just let me know if you have any questions, because I want to clarify those for you. Okay, cool. So let's take a look here at our graph. Okay, so let's say that hemoglobin did not have any cooperativity. Okay, let's say there was no cooperative binding of oxygen to hemoglobin with these different subunits. This would mean we would see when the partial pressure of oxygen in the blood is 25 millimeters of mercury, then we would see about 25% saturation of hemoglobin with oxygen. And if there was no cooperativity here, when we had a partial pressure of 50 millimeters of mercury in the blood, for oxygen, then we would see a saturation of 50% on hemoglobin. Okay, so we would see actually, I'm just going to draw a line here and then erase it. Okay, we would see a linear relationship. That's almost linear. Okay, here we go. We would see a linear relationship between the partial pressure of oxygen and the percent saturation of oxygen on hemoglobin, simply because each subunit would bind oxygen as it became more available, and it would be affected by nothing but that, than the availability of oxygen. I'm gonna go ahead and erase this. Okay, so instead we have that cooperative binding. Okay? We have cooperativity. So what occurs is that and this is a graph we've developed by studying the hemoglobin molecule. Okay? What we see is that, let's say here, at a partial pressure of 40 millimeters of mercury, this would be in our venous blood, we see that, according to this curve and all the research that we've done, we see that and we have to trace our line up, right? To read this, we have to trace the line up, go to the red line, look across. We see that the percent oxygen saturation on hemoglobin is 75%. Okay, so this means when the partial pressure of oxygen is just 40 in our blood, when it's very low, right? The partial pressure of oxygen is 40, that's pretty low for our blood. We still have 75% saturation of hemoglobin with oxygen. This means that in our veins, okay, for the blood in our veins, we have 75% saturation of hemoglobin with oxygen. Isn't that kind of amazing? Okay, the blood in our veins does not have 0% oxygen on hemoglobin. The blood in our veins actually has 3 out of 4 hemoglobin subunits, globin chains in each hemoglobin molecule that are binding oxygen. So we would see this scenario where we have these 4 subunits, right? And we would see oxygen on 3 of them. And an oxygen here. Okay. So this is what our blood actually this is what the hemoglobin molecules look like in our venous blood. That's pretty amazing, okay? So then our blood, right now it's depleted of oxygen. We're only at 40 millimeters of mercury for our partial pressure. Then it's going to travel back to our lungs. Okay? And when this blood arrives at our lungs and gas exchange occurs at the alveoli, our partial pressure of oxygen is going to be very close to 100 millimeters of mercury. And when the partial pressure of oxygen is 100 millimeters of mercury, if we read our graph, we can see that the percent saturation of hemoglobin with oxygen is about 100%. This means that 4 out of 4 globin subunits in hemoglobin are actually binding oxygen in that blood coming away from the alveoli. So this means when we go to the lungs and we pick up oxygen, we go from this scenario right here to adding 1 more oxygen onto each hemoglobin molecule. Okay, so all the time in our body, when our blood is traveling from our alveoli, right, to our systemic tissues, this is how the partial pressure drops. It goes from 100 to 40. Okay, and then when we go from our tissues back to our lungs, we go from 40 to 100. And so we go, and then when our blood comes away from our lungs, we go back from 100 millimeters of mercury of partial pressure of oxygen down to 40. And then when we go back to our lungs, we're taking that deoxygenated blood at 40 millimeters of mercury of partial pressure of oxygen, and we're going up to 100. Okay, so what's happening is every time we go to the lungs, we pick up 1 oxygen molecule on every hemoglobin. Every time we go back to the systemic tissues, we drop off 1 oxygen molecule from hemoglobin. Then we go back to the lungs and we pick 1 up. Then we go back to the tissues and we drop 1 off. Okay, so you might say, like, oh, this seems kind of inefficient. Like, we're only using 25% of the oxygen every time our blood travels from our lungs to our tissues. We only drop off 1 of those oxygen molecules. In a certain way, this does seem kind of inefficient, okay? Because we're only using 1/4 of what we have. But the reason for this, and this is really interesting, okay? The reason for this is because we have these three other subunits right here, these three other globin subunits carrying some oxygen, and this is called our venous reserve of oxygen. Okay? It's basically our backup oxygen. Okay? This right here, it's our reserve oxygen. Whoopsies, something's wrong with my pen. Okay, this is our reserve oxygen. So this is the oxygen that if we get ourselves into some trouble where we actually do not take an inspiration, we do not bring more oxygen into our body for 4 to 5 minutes, we can still survive because hopefully our heart will keep beating and our blood will keep circulating around our body. And as it does, our partial pressure of oxygen will drop lower and lower and lower and lower. Okay. And so for those 4 to 5 minutes, we can continue using all of this oxygen we have right here. We can, as our blood circulates around, one molecule will be gone. Okay, it'll go into our tissues for use. Then another molecule will be gone. It will go into our tissues for use. And another molecule will be gone. Okay. And hopefully, within those 4 to 5 minutes, we start breathing again. Then our partial pressure will go right back up to normal, and we should be fine. So the reason we have all of this extra oxygen that seemingly we do nothing with most of the time is so that we can sustain life if we get into some issue where we can't breathe for several minutes. Okay, cool. So please let me know if you have any questions on this. I know this is a little confusing, especially the first time you learn it. So let's go on to the next slide. Okay, so here we have another graph. This is really showing the same thing. It might look a little bit simpler, and it has some values on the side here. I think I like the other one better, but let me just show it to you briefly here. So same thing, okay? When the partial pressure of oxygen is 40 millimeters of mercury, and this is in our deoxygenated blood in most of our veins. If we read up here, if we read what is the value on the y-axis, what's the percent saturation of hemoglobin with oxygen, and we trace across here, we see that it's about 75% saturated. That means most of the hemoglobin molecules in our blood have 3 out of 4 oxygen molecules bound that they could possibly have. And then when our blood goes to the alveoli and the partial pressure goes up to 100 millimeters of mercury, we can read up here, if we trace up, right, and across, we can see that we have almost 100% saturation of hemoglobin with oxygen. This means 4 out of 4 globin subunits in hemoglobin would have an oxygen molecule bound. So you can kind of see the values here. See how we have when the partial pressure is 100, this is our percent saturation. When the partial pressure is 40, here's our percent saturation. And some other values in here too. So same graph, just slightly different. All right. Let's go into the next slide. This slide is describing, you can see up at the top here, systemic gas exchange. So if you remember in class, we talked about a little bit already alveolar gas exchange. This is the back and forth traffic of oxygen and carbon dioxide across the respiratory membrane in the alveoli, right? Oxygen comes from the air into the pulmonary capillaries, and carbon dioxide goes from the pulmonary capillaries out into the air in the alveoli. On this slide, we're going to talk about the back and forth traffic of oxygen and carbon dioxide at the systemic capillaries and the systemic tissues. So this is where we have the unloading of oxygen, right? Oxygen is actually going to be unloaded from hemoglobin in the blood and cross through the capillary wall to go into the tissue to nourish the cells. And now we're going to have the loading of carbon dioxide, where carbon dioxide actually comes out of the tissues, right? It's a waste that builds up in the tissue. It's going to come out of the tissues and diffuse its way into the systemic capillaries and into the bloodstream. So this is what we're going to examine in this section. So the first thing I want to introduce to you is this chemical equation that we see right down here. This is an equation that is what we call the bicarbonate buffer system. And this equation might look really new for you. I don't know if all of you have seen this before, but I do want you to memorize this. So you need to practice writing it out, section by section until you have all the components memorized. I'm going to explain it to you in detail, and we're going to use it in this video and in the next video to understand acid-base balance. So this is a really, really important equation. So first thing is just memorize it. Okay, so when our carbon dioxide comes into our blood, right, from our systemic tissues, that's what we just said is going to happen. Carbon dioxide is going to be loaded into our blood from our systemic tissues. 90% of that carbon dioxide is actually hydrated, and it forms something called carbonic acid. So that's what we see in this equation right here, okay? The carbon dioxide comes into our bloodstream from our tissues, and it's going to react with a water molecule. Each molecule of carbon dioxide will react with a water molecule. And when it does, we can see what that reaction yields right here. It yields this molecule right here called carbonic acid. So I want you to memorize that this is carbonic acid. And acids, what they do in water, and our blood is essentially water, right? What they do in our blood is they split into two ions. Acids always dissociate. They split into two ions. So see how our carbonic acid undergoes a second chemical reaction right here, where now it splits and it turns into a bicarbonate ion that we can see right here, and a hydrogen ion. So at this point, we have two very different ions, okay, from what we started with here, from these two molecules. Okay, so carbon dioxide, when it's traveling in our blood, it doesn't actually really look like this. Okay, it's not CO2. It's found in this form right here, in this bicarbonate form. Okay, so I'm just going to go over that one more time. Let me erase this. Okay. Once the carbon dioxide comes into our blood, it's going to react with water to form carbonic acid. And carbonic acid, because it's an acid, it splits into two ions, bicarbonate and a hydrogen ion. And this is now what we have found in our bloodstream. So I want to talk about how this shift is driven. Okay, how this shift, where we started with carbon dioxide coming into our blood, and then it reacted with water, and then this equation shifted this way, and then it shifted this way again. So I want to just talk about what is really driving that shift. We would call this a right shift because the equation is shifting to the right. We started off with some CO2 and some water, and then we ended up with a lot of bicarbonate and hydrogen. Okay, so this is called a right shift. I want to look at with you what's actually driving this shift in the equation. So if you remember, what drives the directionality of chemical reactions? Remember, most chemical reactions are reversible. This one is too. So I'm going to draw a second arrow in here to show that it's reversible. So this equation is reversible, and it can shift from the right to the left or the left to the right all the time. And we want to see what's really driving that shift. So if you remember back from AMP1, we learned that, or hopefully you learned, that what drives the directionality of a chemical reaction is the concentration of each of the individual substituents of the reaction. So the concentration of all of these components that we see here, all of these chemical components, will actually drive which way this reaction moves. And if you remember, all the chemical components of a reaction like to be at equal concentrations in a solution. Okay. So this reaction will shift until it finds an equilibrium where there's approximately equal carbon dioxide concentration to water concentration to carbonic acid concentration to bicarbonate concentration to hydrogen concentration. So this reaction will shift back and forth. It will move in one direction, then it'll move in the other direction, until the concentrations of those different substituents that we see there are approximately equal. So let's say we're holding our breath. Okay, let's say we're holding our breath right now. In our body, what would be building up if we're holding our breath? If we're not exhaling, think about it. We would be seeing that right now in our body, if we're holding our breath, we have a little bit of a high concentration of carbon dioxide. Okay. And let's just say the rest of these are pretty much equal. But right now, we're starting to increase the amount of CO2 we have in our body because we're holding our breath. So in order for this equation to come back to equilibrium, because right now it's not in equilibrium, right? It has high CO2. In order to make all of these chemical substituents approximately equal, which way will this shift? Right? If we have high CO2, we want to make this lower, right? We want to get rid of some of this. So the equation is going to shift in this direction, okay? Sending some of this carbon dioxide over to become bicarbonate. Does that make sense? So think about that. If we have high CO2 right now, we need to get rid of some of it. It's too high. The other ones aren't high enough. So we're going to send this CO2 to become some of these other components over here so that we can balance these out, so they can all be approximately equal again. So the amount of carbonic acid will go up a little, the amount of HCO3 will go up a little, and the amount of hydrogen will go up a little, and carbon dioxide will end up coming down slightly, and that's fine. Then they'll all be more approximately equal. Let's do another example. Let's say that right now in our body we are exercising. And when we exercise, we produce a lot of hydrogen ions. Okay. So right now, we have just a little bit too many hydrogen ions in our blood, okay, high concentration. So which way will our equation shift to come to equilibrium? Naturally, on its own. We don't really need anything to make this happen. It wants to do this on its own. We're just going to shift slightly to create an equilibrium. So right now we have a high concentration of hydrogen ions. So this equation will now shift toward the left. Okay? Taking some of this hydrogen, it's going to react with bicarbonate to then become carbonic acid. And then carbonic acid will split into carbon dioxide and hydrogen that we see here. So again, this will balance out all of those different concentrations of those chemical components of this reaction. Okay, so what I want you to do to practice this is I want you to practice writing out this equation, because I do want you to memorize it, and then practice saying, okay, let's say we have high concentrations of water. Let's say we have high concentrations of carbon dioxide. Which way will the reactions shift? Okay. Let's say we have high concentrations of bicarbonate, the bicarbonate ion. Then which way will our reaction shift? So I want you to practice this, because this is kind of the basis for acid-base balance. So if you don't understand this part, it's going to be really confusing to understand everything with acid-base. So start with understanding this equation, memorize it, then look at how does it shift back and forth. All right, so the point I really want to get across right now here to go on in this little section that we're working on is that, is that when carbon dioxide comes into our blood, right, from our tissues, when we're loading carbon dioxide into our blood, we, at those tissues, in those capillaries now, we have a high carbon dioxide concentration, right? We just took a bunch of CO2 from our tissue into our blood. So this reaction, all of these chemical components are always in the blood all the time, okay? They're kind of just hanging out there, waiting until they're needed, and constantly shifting back and forth. So as this carbon dioxide enters the blood, we're gonna have a high concentration. So it's gonna begin to react with water, forming carbonic acid that we see right here. And then that carbonic acid will split to give us the bicarbonate ion and the hydrogen ion. Now, at this point in time, we just generated a lot of bicarbonate and a lot of hydrogen ions. So the hydrogen ions are going to play a really big role in affecting what oxygen does in the body. So let's go down to the next part here. Okay, so now that we've covered carbon dioxide loading from the tissue into the blood, and we've talked about how this carbon dioxide can cause a shift in this chemical equation right here, I want to go ahead and talk about oxygen unloading. So actually look at how the oxygen carried on the hemoglobin in the bloodstream detaches from that hemoglobin and then enters the tissue. So this is actually going to be related to all of these hydrogen ions that we generate when the carbon dioxide enters the bloodstream from the tissue. So that's what we just talked about, how that carbon dioxide, when it enters the bloodstream, because of this equation right here, we see a right shift and we end up generating a lot of hydrogen ions. So these hydrogen ions, they do something very interesting with hemoglobin that's going to help the hemoglobin release the oxygen. So before we get into it, I just want to show you this equation down here. We're going to be looking at this. This is a hydrogen ion plus this right here is called oxyhemoglobin. It's basically where we have hemoglobin that is bound with oxygen. Okay, so this is hemoglobin with oxygen bound to it, 4 oxygens presumably. And over here on the other side of this equation, and this equation is also reversible, although we only have one arrow in here right now. Let's add another one. Okay, this is also reversible. On this side, we have oxygen plus HHB. This is actually what we call deoxyhemoglobin. So I know it doesn't look like deoxyhemoglobin from that little molecular formula right there, but we call this deoxyhemoglobin. It's basically hemoglobin without the oxygen bound, and instead of having oxygen bound, that hemoglobin has some hydrogens bound to it. So the way this is going to work is that because carbon dioxide just entered the bloodstream from the tissue, we're going to end up generating a whole bunch of hydrogen ions. And those hydrogen ions are going to begin binding to hemoglobin. So I'm going to draw over here our hemoglobin molecule we've talked about. And we can see it's 4 subunits there. And let's just put, here we'll use blue. Here's an oxygen molecule in the center of each one. And so this is the hemoglobin we would see in our arterial blood coming into the capillaries. And carbon dioxide would then be entering the capillaries. Okay, so we would see a whole bunch of CO2 nearby. And some of this CO2, remember, has the ability to bind to this hemoglobin molecule. So let's just draw a couple binding here. Okay. But also, other than the CO2 binding to this hemoglobin molecule, remember when CO2 enters the bloodstream, like we have here and here and here, this CO2, we said earlier, causes a right shift in this equation, generating lots of hydrogen ions. And these hydrogen ions can do the same thing as this CO2 over here, binding to hemoglobin, and the hydrogen ions can also bind to hemoglobin. So some of them will bind at certain amino acids around the hemoglobin molecule. And this has the effect of reducing hemoglobin's affinity for oxygen. So when these hydrogens start grabbing onto hemoglobin, when they start binding with hemoglobin, the hemoglobin releases one of its oxygen molecules, usually one, it could be 2. Okay. So one of these oxygen molecules will actually pop right off, okay, will be released because hemoglobin no longer has such a strong affinity for it. These hydrogens here reduce the affinity of the hemoglobin for the oxygen. Now the oxygen is free and it can go right into the tissue. Okay, so this is basically how our oxygen unloading occurs. And the entry of carbon dioxide into the bloodstream and its binding with hemoglobin and the fact that it helps produce those hydrogen ions, which also bind to hemoglobin, that contributes to the ability of hemoglobin to release the oxygen. So this process of carbon dioxide loading and oxygen unloading, those two processes work together. They're kind of like partners. Carbon dioxide comes in, kind of pushes the oxygen away by binding to the hemoglobin and creating hydrogen ions that bind to the hemoglobin. So the oxygen is pushed away and goes into the tissue. Okay, cool. So on the bottom here, we have that reactions that occur in the lungs are the reverse of what occurs in systemic gas exchange. So remember, in the lungs, we have alveolar gas exchange going on. We talked about this in class. And in the lungs, carbon dioxide is going to be unloaded from hemoglobin and exit the bloodstream, go into the air, and oxygen will come in from the air and bind to hemoglobin. So these are simply just the opposite reactions. All right, let's go on to our next slide. Okay, so on this slide, we have 4 factors that adjust the rate of oxygen unloading according to the needs of the tissue, okay? So remember, tissues that are really, really active in our body, they're going to need greater amounts of oxygen. That's simply how they work. They're more active, they need to produce more ATP, and that oxygen is used to produce the ATP. So more active tissues need more oxygen. So we're gonna see 4 ways that the intake of oxygen in the tissue can be adjusted. The first factor here is the ambient partial pressure of oxygen, okay? And this is referring to the partial pressure of oxygen in the tissue itself. So an active tissue, think about an active tissue. Active tissues consume more oxygen. So would the partial pressure of oxygen in there be higher or lower? If that tissue is consuming all the oxygen in there, then the partial pressure of oxygen will be low in a very active tissue. And if, remember, we said the partial pressure gradient between the blood and the tissue is what actually drives the oxygen to move into the tissue. Remember, the partial pressure of oxygen in the arterial blood is about 95 millimeters of mercury. And the partial pressure of oxygen in the tissue is usually about 40 millimeters of mercury or 45 millimeters of mercury, somewhere in that range. So if our tissue becomes very, very active, suddenly our tissue will have an even lower partial pressure of oxygen, making this gradient right here between 95 and 40, or maybe now it's 95 and 37 millimeters of mercury, will make that gradient even steeper. So oxygen will be diffusing faster into the tissue. So the second, let me erase that. The second factor adjusting the rate of oxygen unloading is temperature. Active tissues promote greater oxygen unloading. Tissues that are warmer, if it has an increased temperature, then we get greater oxygen unloading. And I want to show you a little graph to show you why this happens. Let's go on to the next slide. So on this slide, we can see how temperature affects the oxyhemoglobin dissociation curve. So I just want to point out to you here, 38 on this graph is about the most normal temperature we have on this graph. Okay, so we're going to say that this line right here, this oxyhemoglobin dissociation curve, is the normal one. And so here at a partial pressure of 40 millimeters of mercury, we can see that our hemoglobin is about 75%. Oh, that looks more like 70 on this graph, but somewhere in the 70 to 75% range of saturation. And here at 100 millimeters of mercury of the partial pressure of oxygen, we have close to 100% saturation of hemoglobin with oxygen. Okay, so this was just like the first graph that we looked at. So now let's just see if we increase our body temperature. Now our body temperature wouldn't increase this much, okay, but we can see that there's a trend that our curve would move over to the right. We would see a right shift to the curve if we have a higher body temperature. So we're just going to use this line as an example at 43 degrees Celsius. Okay, let's see what happens with the oxyhemoglobin dissociation curve. So at a higher temperature, now at a partial pressure of 40 millimeters of mercury, so like right here, right? If we trace that line up, it's about right here. Look at now, our percent saturation of hemoglobin with oxygen is at about 50%. So this means in the body where the partial pressure of oxygen is 40, right, like at the tissues, let's just do this, okay, like at the tissues, then 2 out of 4 oxygens will leave hemoglobin. Okay, if we're at 50% saturation, that means 2 out of 4 oxygens will leave hemoglobin. So in a warmer tissue, like let's say we're exercising, right, and our muscles are heating up, and so we're producing a lot of heat in this area. So when the blood circulates into this area, it warms up, and the hemoglobin now releases its oxygen even easier. Okay, it releases it just a little bit quicker because we are increasing this temperature. This graph, we produced this graph based on the properties of hemoglobin. So we've studied hemoglobin and we've seen that at a higher temperature, well, it releases its oxygen more quickly at a lower partial pressure than it does at this normal body temperature. Isn't that kind of cool? So just because the tissue is warmer, it releases more oxygen. That's just the way the protein works. Okay, let's go on here. Back to this slide. Okay, so the third factor affecting oxygen unloading is called the Bohr effect. And I've actually just explained this to you on the last, one of the last 2 slides. So the Bohr effect is that an active tissue promotes oxygen unloading because of an increased hydrogen ion concentration and a low pH. Okay. So a very, very active tissue, not only will it be using up more oxygen, but it's also going to have high CO2 levels, right? Because tissues, one of their waste they produce is carbon dioxide. So if we have a really active tissue, they're going to be producing a lot of carbon dioxide. And remember on the last explanation, on that one of those last slides where I was saying carbon dioxide will go and bind to hemoglobin, and it will actually reduce hemoglobin's affinity for oxygen. And then we also said hydrogen ions can do the same thing, right? So when we have a lot of hydrogen ions in a tissue, a very active tissue also tends to produce lots of hydrogen ions. And we also said whenever we have more carbon dioxide, we're going to have more hydrogen ions because of the shift in the bicarbonate buffer system equation. So these hydrogen ions can also go and bind all over hemoglobin, causing it to release more oxygen. Okay, and so when more oxygen is released, more oxygen will go into the tissue and nourish that tissue. Another thing we see is that at a low pH, right, and this is related, at a low pH, hemoglobin behaves differently. Just like in this last example where hemoglobin behaved differently at a higher temperature, we're going to see that at a low pH it also behaves differently. Let's go look at that graph. On this graph, we can see that at this pH right here, okay, at a pH of 7.4, this is about our normal physiologic pH, right? 7.35 to 7.45 is our normal range for pH. We can see our normal curve. Okay, this is just like what we saw before. At a partial pressure of 40 millimeters of mercury, like we have at the tissues, our percent saturation of hemoglobin with oxygen is about 70 to 75%. So great. Now, if we just lower the pH just a little bit, okay, to 7.2, see how much our curve shifts to the right? So now at a partial pressure of 40 millimeters of mercury, our hemoglobin population is only 60% saturated with oxygen. So this means more molecules of hemoglobin will release 2 molecules of oxygen when they get to the tissues. Because if the tissue is more acidic, right, has a lower pH, like active tissues do, then more oxygen will be unloaded. And that's perfect because that tissue needs more oxygen right now. Okay, just like in those last 2 examples where if our tissue is warmer, that means it's more active, it needs more oxygen now. And also if our tissue is using up more of its oxygen, it's also going to need more to replace it. So these mechanisms work perfectly to provide the tissue with exactly what it needs. Okay, and then finally we have our last factor affecting oxygen unloading, and that is down here involving a molecule called bisphosphoglycerate, or BPG. This is a molecule that is part of a process called glycolysis that we're going to study later in the semester. It's one of the intermediates in this long process of breaking down glucose. But red blood cells are some of the cells in our body that produce a lot of BPG. Okay. They produce a whole lot of BPG. And BPG can bind to hemoglobin, similar to the way carbon dioxide and hydrogen were binding to hemoglobin on that last slide. And when BPG binds to hemoglobin, just like when carbon dioxide and hydrogen do, the oxygen is unloaded more quickly. So binding of BPG to hemoglobin reduces hemoglobin's affinity for oxygen. So oxygen is released. Now also, when our metabolic rate increases in our body, okay, when we have an increased metabolic rate, this requires our body to produce more oxygen. Now, also, if our metabolic rate is higher, our body will produce more BPG in our red blood cells and in other cells in our body. A great example of this would be when we have a fever. Okay, so if we have a fever, our body has a higher metabolic rate because all of our chemical reactions are occurring faster. So we're going to need more oxygen to produce more ATP so we can power all of those chemical reactions that are occurring faster now because of our temperature. So one of the things we see is that our cells, they produce more BPG during the fever. Okay? And so BPG, we said, can bind to that hemoglobin and cause that hemoglobin to release its oxygen with more ease. It reduces that hemoglobin's affinity for oxygen. And this is perfect for when we have a fever because since we have more BPG during the fever, we'll get to release more oxygen. That oxygen can go into our tissues and help us heal faster so that our fever can then go away. So all of these factors are allowing our tissues to basically regulate their own access to oxygen, okay? Each tissue, just by producing extra carbon dioxide or having a low amount of oxygen in it or being a little bit warmer, can regulate its own oxygen supply. Because just by increasing that temperature, that's going to cause the hemoglobin to behave differently. Just by decreasing the partial pressure of oxygen, that's going to adjust the rate at which oxygen diffuses into the tissue. So these mechanisms have evolved to make us the most efficient and make our hemoglobin the best molecule for delivering oxygen to our bodies because the hemoglobin behaves differently under these couple of different conditions here. Okay, cool. So let's go on. All right, this is a quick little slide. I don't want to spend too much time on this, but I just want to point out to you that our fetal hemoglobin and adult hemoglobin behave differently. Okay, so here's the curve for adult hemoglobin. We can see it right here. It's the same one we've been looking at this whole time. We're at a partial pressure of 40 millimeters of mercury. We have about 75% oxygen saturation on hemoglobin. Now, the fetal hemoglobin, notice this, okay? At a partial pressure of 40 millimeters of mercury, okay, the fetal hemoglobin, let's trace this up here, the fetal hemoglobin has a saturation of 90%. Okay, so this is about 75% versus 90%. This means at the same partial pressure, right, at 40 millimeters of mercury of oxygen, the fetal hemoglobin has a greater affinity for that oxygen than the adult hemoglobin does. So if both of these hemoglobins are together in the same solution in the body, or even very close to each other in two different mixtures in the body, or two different solutions in the body, if they're both given the chance to grab oxygen at a given partial pressure, let's just say 40 millimeters of mercury, let's say that the partial pressure of oxygen in this fluid is 40 millimeters of mercury, the fetal hemoglobin will grab more of the oxygen than the adult hemoglobin will. And this is really, really important in our body because the mother's blood will pick up the oxygen in her lungs. The mother's hemoglobin will pick up the oxygen in her lungs, and it will transport it to her placenta, where the mother's hemoglobin then needs to hand off the oxygen to the fetal hemoglobin. And this would only work if we have this scenario that we have right here, where the fetal hemoglobin wants to bind to the oxygen more than the mother's hemoglobin does. So basically her hemoglobin can pass the oxygen off to the fetal hemoglobin. So I think this is kind of cool. I'm not going to test you on this one. I do want you to know about the other graphs But this one I'm just kind of telling you about for your own interest. Okay, so the last part of this lecture, and I know this is a really involved piece of the material, even though it's only about 15 slides long. But it is quite a complicated section in the material. So take your time on it, okay? All right, so the last section here is about blood gases and the respiratory rhythm, and then we're also going to talk a little bit about acid-base balance in this section. So first thing that I want you guys to know is that, do you remember the respiratory centers in the brain, the VRG, ventral respiratory group, the DRG, dorsal respiratory group, and PRG, pontine respiratory group? They are the regions in our brainstem that are regulating our breathing rate and depth. The DRG does this to maintain levels of these three chemicals right here. When we talk about pH, we're actually talking about hydrogen ion concentration. Talking about the concentration of hydrogen ions. And we need the pH of the blood to be between 7.35 and 7.45. So first thing the DRG is monitoring is that, okay, this pH needs to be in this exact range, otherwise we need to change the rate and depth of breathing to make sure we maintain this exact pH range. And then we also want to monitor our partial pressure of carbon dioxide. In our arterial blood, this should be 40 millimeters of mercury, or right around there. And the third thing we want to monitor is the partial pressure of oxygen, which should be right around 95 millimeters of mercury in our arterial blood. These levels will obviously be different in the venous blood. So this is just for the arterial blood. So I want you to memorize these, first of all. These have to be memorized, okay? And then our, the DRG is most sensitive to pH changes, then carbon dioxide changes, then oxygen changes. Our most potent stimulus for breathing is pH, followed by CO2, and the least significant one is O2, okay? So we're mainly monitoring that pH. Now, do you remember that our chemoreceptors are the ones that monitor these three factors here? Okay, so the chemoreceptors are monitoring these. Remember, we have them in the central nervous system, in the medulla, and they monitor these values in the cerebrospinal fluid. And then we also have peripheral chemoreceptors in the internal carotid artery and in the aorta that monitor these values in the blood. Okay, so make sure you know that. I know we've talked about that before, so that should be a review for you. On to our next slide. Okay, so the first thing I want to do on this slide is talk about pH for a moment. So I want to talk about when our blood is acidic, what does that mean? And when our blood is more basic, what does that mean? And actually, I shouldn't say that our blood is acidic, technically, okay? The pH of our blood is close to neutral. It is just slightly basic. But when our blood becomes more acidic, okay, or when our blood becomes more basic, I want to talk about what that means. So our blood should be between 7.35 and 7.45. And we said that on the last slide. Now, if the pH is below 7.35, then we are considered to have a condition of acidosis, okay? So this would be a low blood pH. When our blood is more acidic, that means we have a low blood pH. Okay? When our blood pH is higher than 7.45, we are considered to have a condition of alkalosis. So when our blood is basic, this means we have a high blood pH. Now we need to know what this means for our hydrogen ion concentration in our blood. And this is really, really important. So if our blood is acidic, this actually means that we have a low pH but a high concentration of hydrogen ions. And I know that that might not be intuitive for you to memorize, so I just want you to memorize it, okay? So acidic means that we have a lower pH but a higher concentration of hydrogen ions. I like to think hydrogen ions cause something to be acidic, okay? So when we have a lot of hydrogen ions, that means we're becoming more acidic. And if our blood has a higher pH, higher than 7.45, this means we have a low concentration of hydrogen ions. So you do need to memorize this, because if you get this confused, this will make answering questions about blood pH really, really difficult for you, because you'll constantly be second guessing yourself. So you need to be really confident about this. So I would just absolutely memorize this so you don't have to question yourself at all when you answer questions about it. Okay. Now, there's a couple of other terms here. Okay, so the first term I want to talk about is hypocapnia. Okay, so hypo, you guys all know this means a low. Okay, and capnia is referring to our carbon dioxide level. So hypocapnia is a low level of CO2. It's a partial pressure of CO2 less than 37 millimeters of mercury in our arterial blood. So this means we have a very, very low carbon dioxide level. Okay, and it says down here that this is the most common cause of alkalosis. So we need to think about, why would this be? Okay, why would this cause alkalosis? So I want to go ahead and write out this equation down here so we can revisit it. All right, so I just decided to go ahead and type it out on the slide. I thought it would be a lot easier and clearer for you to read. So we have our equation here. This is the one we were discussing earlier. Remember, this is called this is the chemical equation for the bicarbonate buffer system. And so the reason that hypocapnia causes alkalosis is found in this equation right here. Okay, so let's think about this. So if we have hypocapnia, we said that's a low CO2. So we've got a low, I'm just going to use a down arrow for this. Okay, we've got low CO2. And this means that we don't quite have enough CO2 in our blood, and maybe we have more water in our blood, more, what is this again? Carbonic acid. Okay, maybe we have more, what is this again? The bicarbonate ion, and maybe we have more hydrogen ions in our blood. Okay, so the other components of this equation may be at higher levels in the blood because now we have low CO2. Okay, and remember, this equation will balance itself out. It will adjust, it will either shift over to the left or to the right in order to balance itself out and make all of these different components equal to one another, make their concentrations as equal as possible to one another. So because we have low CO2, okay, you have to think about which way will our equation shift to bring this CO2 level up a little bit. So to generate more CO2, our equation is going to shift over to the left. It's going to shift over to the left. It's going to take some of this hydrogen and these bicarbonate ions, convert them into carbonic acid, and then carbonic acid will generate water and carbon dioxide. Okay, so does that make sense? So because we have a low CO2 level, the equation is going to shift to push some of these other components over to the CO2 side. So we generate some more CO2, bringing that level up a little bit higher and decreasing the level of those other components. Okay, so now we've pushed this equation over to the left. Great, okay. We have some more CO2 now, but how does this cause alkalosis? Remember, what is it when we have alkalosis again? Okay, it says here, alkalosis is blood pH higher than 7.45. So let's look down here. When we have a high pH value, this means we have a low hydrogen ion concentration. So if we look back at this equation, how do we have a low hydrogen ion concentration in this situation right here? Okay. Remember, we just shifted some of these hydrogen ions over to the left to allow us to replace some of that CO2. Okay. So we end up with a very low hydrogen ion concentration. This is what causes our alkalosis. Okay, cool. Let's talk about the next one. So let's get another color here. Okay, so for hypercapnia, okay, hypercapnia. In this case, hypercapnia, high levels of CO2. This is where we have a partial pressure of CO2 greater than 43 millimeters of mercury. Remember, it's normally about 40. So this is our most common cause of acidosis. So let's see why, okay? So if we have a high CO2 level, remember our partial pressure of CO2 is high. Now CO2 is a little bit higher than all of these other components. These guys all have a slightly lower concentration than the CO2. So this equation is going to shift to balance them all out and make them all approximately equal. So to do that, this equation will shift to the right, pushing some of that CO2 over here to this side, causing the number of our hydrogen ions to increase, causing an increase in our hydrogen ion concentration. So because we have a high hydrogen ion concentration, this means right here, right, high hydrogen ion concentrations, this means our pH value will get lower, right? Maybe our pH will drop below 7.35 and we will generate an acidic condition in the body. So I really want you to spend the time and think about how this works. Okay? I want you to use this equation, memorize this equation, memorize how it shifts if you need to. Say, okay, if I have a low CO2 levels, if I have hypocapnia, what will occur? What will happen with my hydrogen levels? Will my hydrogen ion levels increase or decrease? Will my pH value increase or decrease? And then give yourself another example. Say, what if my bicarbonate levels are a little bit too low? Then what will happen? What if my hydrogen ion levels are too high? Then what will happen? Okay, so really practice with these so you can get the hang of how this equation shifts and how that affects our body's pH. All right. Another trick you can use for this, and this is just a trick, it's not a full understanding of this, but as like a quick way to memorize this, you can memorize whenever our CO2 levels are high, that will cause our hydrogen ion levels to become higher, okay, to increase as well. And whenever our CO2 levels are low, I'm sorry, my pen is not working right now very well, okay? That will, you should just write this in your notes though, okay? That will cause our hydrogen ion levels to also be low, okay? So if our CO2 levels go up, hydrogen levels go up. We can see that right down here, right? When our CO2 levels increased, our equation shifted to the right, causing our hydrogen ion levels to increase. The opposite is also true. You can see it in the last example. When our CO2 levels were too low, that caused our hydrogen ion levels also to decrease. Okay, so that's one little trick you can use, but you still have to understand if we have low hydrogen ion levels, what does that mean about our pH? Well, that means our pH value is high. What is that? Acidosis or alkalosis? That's alkalosis. So you do have to memorize that part. Okay, cool. So let's go ahead and go on. One thing I forgot to mention earlier is that there's an enzyme that actually catalyzes this step right here, where carbon dioxide and water join to form carbonic acid, or the reverse, where carbonic acid splits apart to form carbon dioxide and water. There's an enzyme that catalyzes both of these reactions, and that enzyme is called carbonic anhydrase. And this enzyme speeds up this reaction. Remember, enzymes, they don't just cause the reaction to occur, they actually speed up the reaction. The reaction would occur on its own. The enzyme just allows it to occur much faster. So that enzyme is present in our bloodstream, and it can catalyze the forward reaction and the reverse reaction just at this step right here. So I do want you to know the name of that enzyme as well. All right, so this slide talks about respiratory acidosis and respiratory alkalosis. This is essentially what we've been talking about. It's pH imbalances that result from a mismatch between the rate of our pulmonary ventilation, right, how quickly or slowly we're breathing, and the rate of our CO2 production. So if we're producing a lot of CO2 in our body and we're not breathing fast enough to expel it, the CO2 will build up in our body. That will then increase the concentration of hydrogen ions, causing us some acidosis. So we want to make sure that our CO2 production is matched by our ventilation rate. And when it's not, we're going to develop one of these conditions here. Okay, so let's look at this down here. This says that hyperventilation. Do you guys know what hyperventilation is? Right. I'm sure most of you have either experienced this or seen someone experience it, or you can even get what it is from the name. So hyperventilation is a high ventilation rate, right? It's very, very fast breathing. So this is actually, in our body, it's a corrective homeostatic response to acidosis, okay? So remember, if we have acidosis, right, that's a high concentration of hydrogen ions. If we have a high concentration of hydrogen ions, we just saw on that last slide that we also have a high level of CO2. So a way we can correct this is by breathing extra fast to expel a whole bunch of that CO2. Okay? And as we're pushing that CO2 out of our body, okay, if this is our equation here, right now we're decreasing our CO2 levels by pushing it out of our body. This equation will actually shift over to the left, and we've seen this on the previous slides. Taking some of this hydrogen and bicarbonate from over here and moving it over to the left, decreasing the amount of hydrogen in our bloodstream. And this is good, right? Because we just said our problem was we had too much hydrogen in our bloodstream. We had acidosis. So this corrects our acidosis. By expiring that carbon dioxide, we correct our acidosis. We bring down our hydrogen levels, and so our pH value will go back up to normal. Okay, this raises our blood pH back toward normal. Okay, so make sure you understand this. If you need to go back and listen to this again, go back and do it right now. On this slide, we have that hypoventilation is a corrective response to alkalosis. Okay, so if we have alkalosis, this means we have a low level of hydrogen ions in our blood to begin with. Okay, so this is what we're starting with, a low level, a low hydrogen ion concentration. And we said if we have a low hydrogen ion concentration, we also have a low CO2, or a low partial pressure of CO2 in our blood as well, and a low level of carbon dioxide. So we need to correct this. Okay, so what we're going to do is we're going to hypoventilate. We're going to breathe slower. If we breathe slower, we actually expel the CO2 a little bit slower. This allows the CO2 to accumulate in our body. Okay, so we're just breathing slow, so the CO2 kind of builds up in our body. So all of a sudden, let me use a different color arrow here, we increase our CO2 levels because we're letting it build up in our body by breathing slower. That's going to cause our equation to shift over to the right, pushing some of the CO2 and water over on this side, increasing our hydrogen ion levels. Okay. So this brings our hydrogen ion levels up. Okay. So this means our pH value actually goes down. The value actually goes down. So our blood becomes less alkaline, goes back toward normal. Maybe it goes from 7.46 to 7.4. Okay, so it's actually going to drop that pH value. And we increase the concentration of hydrogen ions. Okay, so that's exactly what that says here. It raises our hydrogen ion concentration and it lowers our pH to normal. So if this isn't making sense to you, Okay, well, first of all, I hope this is making sense to you, okay? And if it is, regardless of if it is or if it isn't, I want you to practice this a few times on your own when you're studying. I know the study guide is designed to take you through this, but I really want to emphasize to you how important it is to practice this, because if you don't practice it, it's going to be hard to get it right when it comes time for the test. And then if this isn't making sense to you, I want you to go back and listen to it again. And while you're going through this, I want you to write out the equation and imagine the different chemical components. Imagine, okay, if these increase over on this side, how will they shift? How will that affect, if we're changing carbon dioxide, how will that affect our hydrogen ion concentration? Okay, so I want you to write it out in your own notes. Almost done here. So our pH, what we just talked about, our pH has huge effects on our ventilation rate, because pH is the thing our brainstem is most sensitive to, and that those chemoreceptors are most sensitive to. Now, our carbon dioxide levels also have effects on our respiration rate and depth. They have indirect effects through pH, as we just saw, right? Whenever our carbon dioxide levels change, that affects our pH, and then our chemoreceptors detect that and cause our breathing rate to change, or our breathing depth to change. But carbon dioxide also has direct effects on breathing. So when we very first begin our exercise, we might have a slightly elevated CO2 level. We might have a high partial pressure of CO2, because when we first start exercise, our breathing rate hasn't increased much yet. This is like in the first minute or so of our exercise. And our muscles are working harder, so they're producing a lot of CO2, yet we haven't begun to expel it very quickly yet, because our breathing rate hasn't increased. So this high CO2 level can actually stimulate our peripheral chemoreceptors, right, those in our aorta and internal carotid artery, or arteries, and trigger an increase in ventilation more quickly than our central chemoreceptors can in our brain. So those chemoreceptors might detect that high CO2 level and signal our brain to increase our ventilation rate. So let's go on. And then on this slide, we have that our partial pressure of oxygen usually has very little effect on our respiration rate or our ventilation rate. The only time we really see our oxygen level affect our rate or depth of breathing is if we have chronic hypoxemia. Okay, so all the time we have a low blood oxygen level for some reason. This would cause our partial pressure of oxygen to be very, very low, okay? Maybe less than 60 millimeters of mercury. When this happens, our chemoreceptors detect this, and they tell our brainstem to significantly cause an increase in our ventilation depth and sometimes rate as well. So the effect we see, once our brainstem kicks in, if we have this very low partial pressure of oxygen and our brainstem kicks in like, okay, we got to get some more oxygen in this body, we go into a condition, what we call here hypoxic drive. And this is where our respiration is driven more by a low partial pressure of oxygen than by carbon dioxide levels or than by our pH. And we see this if a person has emphysema, where they're literally just not able to take in enough oxygen in their lungs, or again, if they have pneumonia, if they have a little extra fluid in their lungs. Both of these can interfere with gas exchange, alveolar gas exchange in the lungs. So the person will just be getting a much lower amount of oxygen crossing their respiratory membrane. And this will cause them to go into hypoxic drive, where breathing sounds very labored and like they're using a lot of effort to pull in air into their lungs, simply because these conditions are inhibiting their gas exchange, their alveolar gas exchange. And then we can also see this condition if we've been at high elevations after several days. So again, we have this low partial pressure of oxygen. So our body's response to that is to cause this very deep and labored breathing to occur so that we can attempt to bring more oxygen into our body. All right, and then The final thing I want to talk to you about here is respiration during exercise. So the reason our ventilation rate increases during exercise is because our brain is sending motor commands to our muscles to increase our movement, okay? And so our muscles are moving more, all right? And so because consciously in our brain we've made a decision to move our muscles more, at the same time our brain is going to send signals down to the pons and then further down into the dorsal respiratory group in the medulla oblongata and tell those regions of the brainstem, hey, our muscles are moving more now. We're going to need some more oxygen soon, okay? So in anticipation of increased oxygen need, our brain is actually increasing our ventilation rate. We don't want to begin exercising without beginning to increase our ventilation rate and then continue exercising so that in our body we get so low on oxygen that we pass out, okay? We don't want that to be the scenario. So our brain will sort of preemptively send signals to the medulla saying, hey, we're starting to move our muscles more now. We're sending more signals to the muscles. Let's also increase the ventilation rate before our oxygen levels get too low. Also, the proprioceptors that we have, these are the little receptors in our muscles and joints that can sense how much our muscles and joints are being stretched and moved. They will send signals to the respiratory centers in the brainstem, and those signals will stimulate the brainstem to cause an increase in the breathing rate. So same kind of idea. Our muscles are moving more, so we're sending signals from those little proprioceptors in the muscles up to the brainstem saying, hey, we're going to need more oxygen down here soon. Let's increase the ventilation rate. All right, so let's go ahead and end there. And I just want to remind you guys that we will have a quiz on this. My Santa Fe class will have our quiz on the Monday after spring break. That's in 5 days. And I hope you guys are having a lot of fun on your spring breaks. I know I've been pretty busy. This is the second day I've been filming this video, and it has been very, very difficult to teach this via video instead of in person. I don't know exactly why. I think it's because it's harder with the tools I have in front of me and with the computer. So I really, really appreciate your patience with this. And I hope you're having so much fun or just getting so much rest and relaxation. And I can't wait to see you in about 5 days. Okay, cool. So thanks for watching, and I'll see you next time.